Now, the last part of this chapter deals with mutations. Mutations are changes in the genetic code. These are errors made when the cells are copying their DNA, when things are done incorrectly. And mutations have the most impact when they occur in cells leading to sperm or egg, because if a sperm or an egg has a mutation in it, then every cell of the organism produced by that sperm and egg will have that mutation. Meanwhile, if you get a mutation in your big toe, that's not going to be passed on to the next generation. It might impact you. It could develop into cancer or something like that, but it won't be passed on from generation to generation. But we're going to focus on small-scale mutations today. So big mutations are when like a whole chromosome gets copied by accident. You get extra copies, or you add on a whole piece to that chromosome. Those are big mutations. We won't worry about those today. We're going to talk about small-scale mutations, focusing primarily on point mutations, which are changes to one set of bases. So like, let's say we have a double helix of DNA, it's CGC. Then on the other strand of DNA that's complementary to that, that would be GCG. Well, what if instead of GCG, these got flipped and it was CCC and this was GGG? That would be a single change. That would be a small scale mutation, or a point mutation. So point mutations are a type of small-scale mutations, and they're going to include substitutions and insertions or deletions. Substitutions is when you have the wrong bases there, and insertions or deletions are when you delete one set of bases. And that can lead to a bigger kind of mutation known as a frame shift mutation, which we have a diagram to show you that in a moment. Okay, so let's first look at a basic substitution. Okay, so here we have hemoglobin. This is this is a red blood cell, an RBC, a red blood cell, and this is one as well. And hemoglobin is a protein found inside of red blood cells that carries oxygen. And in wild type is the name given to the kind of gene found in most individuals. Okay, so wild type. In wild type individuals, we have this DNA sequence. This is the genotype, and this is the phenotype. This is what the red blood cells look like. Sorry about the line. Phenotype, this is the genotype, right? The kind of genes they have. When this gets transcribed to RNA, we have this, and then you can look this up. GAG. See how it says GAG is the codon? If you go back up to, where'd it go? There we go, right here. Let's look up GAG. So GAG right here. GLU is the protein, or is the amino acid that GAG codes for. GLU is what is coded for there. So let's go back. Should have put that closer to this slide, eh? All right, so GLU is the protein. Okay, so is the amino acid. There's another amino acid here and another one here, but we're not worried about those. We're worried about this one. So what this is showing down here is that primary structure of the protein, the different amino acids, and we're focusing on this one. Now, in some individuals, there is a substitution. These have flopped. Notice it's CAC instead of CTC, and this is GTC. So basically, these have changed places. And this is the mutant type of the DNA, and it codes for GUG. And if you look up GUG on that list, it codes for VAL instead of GLU. Okay, it's valine instead of glutamic acid. Now, this is important because glutamic acid, I think I'm getting this right. This is hydrophilic, meaning it likes water, while this is hydrophobic. So when it's hydrophobic, that means that part of the protein turns away from water. It changes the shape of the protein. And then when you have a whole bunch of those proteins interacting in the cell, they interlock in a different way. They cause the entire cell to be a different shape. So we have a whole series of emergent properties here. We change one amino acid, or well, we change one nucleotide in the DNA, which changes one nucleotide in the RNA, which changes one amino acid in the protein, changes the shape of the protein, which changes the shape of the entire red blood cell. And now these sickle-shaped cells can get stuck in capillaries and cause heart problems or cardiovascular problems, and can lead to early death of the organism. So people, in this case, human beings, suffer from this condition. Now, this can actually be a useful trait as well. If you only have one copy of this gene, you do have some sickle cell symptoms sometimes, but you can also fight malaria better than people who don't have it. So it's an interesting tension between usefulness and causing disease. But we'll talk more about that when we talk about Gregor Mendel and humans. All right, so let's look at the different kinds of mutations. So we have a really nice example here from the book. I was going to make my own version of this, but the book's version is better than I can make. So here we have the DNA strand. We have the double helix shown here, both strands, and we have the messenger RNA strand. Okay, and we have the protein, the polypeptide produced here. It's a really short one to make it easy to understand. MET, LYS, PHE, and GOI, those are the amino acids, and they're all stuck together, and that's what it looks like. Okay, there it is, all done. Now it's going to show you various kinds of mutations. Okay, so there are different consequences of nucleotide substitution, so this type of point mutation. So here's an example of a change. Here we have A, whereas up here we actually had a G right there, but now we have an A, so an A and a T. Now, this is going to result in a mutation known as a silent mutation. Why? Well, if you look here, this used to be GGC, and now it's GGU. Okay? It was GGC, now it's GGU. Let's look at what happens. GGC, let's find GGC. Help me out. Do you see it? Does anybody see it? Somebody point at it for me. There you go, GGC. All right, it codes for GLY. But now we've mutated to GGU, but it also codes for GLY. So we've had no change in the protein. So because there's no change in the resulting amino acid, this is known as a silent mutation. So this is no change in the amino acid. Okay? So we've changed the DNA, we've changed the RNA, but it hasn't affected the amino acid. Why? Because the genetic code is redundant. Codons are redundant. There's more than one codon for each amino acid. So there's a little bit of a safety net there. Okay, now here's another kind of mutation then. Let's say now instead of a C here, we have a T here. So T and A are here, right? T and A are right there. This is known as a missense mutation. This is the same kind of mutation we saw in sickle cell. This is when you end up with a new amino acid. Or you might think of it as the wrong amino acid is coded for here. And what I would like you to do is to look this up. Figure that out, look it up, and figure out, oh, here's why it changed. Look it up on that table. Now, to do that well, what you can do is divide each codon up to make them easier to see, like that, and that will make it easier to look up. Okay? So you can see what the codons are. All right, the last kind of mutation that's going to result from this small scale substitution is a nonsense mutation. Nonsense mutations can have quite a bit of an effect on a protein, especially depending where it is. If it's towards the end of the protein, it won't matter as much maybe, but it can be pretty bad. Nonsense mutation is when you go from an amino acid to a stop codon. So instead of an amino acid there, so it used to be, let's see here, this is the second codon. It used to code for LYS, but now it codes for stop. So what would actually happen? Notice what actually happened here. Only the first amino acid is put down and none of the rest of the protein is even manufactured. So you had a massive change to that protein because as soon as you hit stop, everything comes apart and you don't continue production. Okay, those are some small scale a type of point mutation known then as substitutions, and their specific effects. You can have nonsense, you can have missense, or silent. The second issue you can have comes from insertions and deletions. And once again, I think what you ought to do is draw a line here, here, here. Oh, did I do that right? Yeah, okay. And here. And let's refer to each of these boxes as a frame, kind of like a picture frame, where you can see the codons right there. You can see what they are, right? Almost like a picture frame frames a picture. These frames frame every three nucleotides, or every three nucleotides, letting us see the codons. Now, when we have insertions or deletions, that causes a massive impact, because an insertion or deletion changes the frame. What I'd like you to do is draw the frames now on this one. We have had an extra A and T inserted here, so here's the new messenger RNA. So the first part is right. What happens next? I'll let you figure that out. But you can see what happens. But once again, kind of like a nonsense mutation, suddenly a stop mutation has appeared here, and so everything stops. That's a frame shift mutation leading to a nonsense mutation. But they call it a frame shift mutation because it's like we shift the picture frames. They shift to the right or to the left, changing all kinds of things. So these can have massive changes to a protein, especially if they happen early on in the protein. If you have a deletion or insertion early on in that gene, it's going to cause a massive change to that protein. All right, let's look at another one. Here is a frame shift mutation. Here is an A deleted, and that causes all of these amino acids to change. That is going to be a missense mutation caused by a frame shift. And then lastly, we have here in this case they have three. They deleted a whole codon here, which doesn't change the protein as much in this case because you've deleted an entire frame instead of just one base. But none of this is going to make sense. Let me just warn you, I'm just talking to you about this, and I'm not meeting you in person while I'm making this video. So if you're not seeing me this semester in person, none of this will make sense until you get out a piece of paper and you write this down and you do it. Okay? You have to do it. It's not a game of the mind, it's a game of physically doing this until you understand it. And it's understanding how to solve these problems. So work on the examples in the book. Do the examples that you might see online, but I also have a worksheet for you to work on as well. So if you delete all three, like in this case, we might wipe out all three of these and delete them. In that case, some of the protein's still the same. We still have these two down here, but we still have two of the amino acids that we originally had, but we're missing one now. So some things might stay the same after that if you've removed all three of that codon. But like I said, that won't really make sense until you play with it. So figure that out. But just stepping back out again, just bear in mind that we've taken DNA, we've opened it up, we've made messenger RNA, we've edited messenger RNA, it's exited the nucleus, it's gone to a ribosome, and then we've built a protein, and that protein is going to go off to do all kinds of different jobs depending on the protein in the body. The worksheet I've made for you looks something like this. It's going to part of it's going to have a table like this, and I gave you an example that's filled out already. It's got the DNA sequence, the messenger RNA sequence, and the amino acid. So I want you, and there are more questions than that on there. I'm going to have you do this and turn it in. So if you have any questions on that, be sure to contact me. Now, the last thing I want to mention then, before I go, is that mutation is the source of new genes. A lot of times it results in silent mutations or a problem that leads to disease. When a mutation causes disease, it's called a genetic disorder, right? Or an inherited disease. but sometimes it can result in a slight benefit. And natural selection works when there is variation in the population, like we see in this ladybug population. That variation came from mutation, different mutations leading to different color patterns. Now, perhaps some of those color patterns are more camouflaged than others. Those individuals then that are more camouflaged will have more babies that survive. So more offspring are produced in a population than will survive till adulthood, whether it be a fungus making spores or baby birds. But some of them will survive more than likely, and usually the ones that survive happen to have traits that make them a little better adapted to their environment. And then they go off to have their own babies. So because of that, populations over time adapt to the environment. And none of that would be possible without random errors leading to slight variation. It's almost like an experiment. Which one of these will work better in this habitat? We'll learn a lot more about evolution in Bio 2. But that's the basic premise here. And that leads to over time to all of the diversity of life that we have on this planet. This is just less than 1% of known species shown on an evolutionary tree. And right there is humanity. We are part of this amazing web. All creatures are our cousins, and I think that's pretty awesome. All right, we're going to move on then. This is the end of week 1 if you're taking the class in summer, and we are going to move on very soon to an exam, and we're going to move on as well to learning about mitosis, meiosis, and other exciting things. So, hopefully you've stuck with me, and hopefully this has made sense. And I will see you soon.